Good evening, good afternoon, ladies and gentlemen. The third lecture is this year scheduled in the afternoon, and it is again a great pleasure to introduce to those of you who may have missed first two lectures of this year's Pauli Lecture series, Professor James Rothman. He's visiting us from Yale. He's there a professor and the chairman of the Department of Cell Biology. And his research has made a profound impact on our understanding of vesicle traffic, and it has been recognized. And Professor Rothman received numerous awards and prizes, among which the Lasker Basic Science Award and the Nobel Prize in Medicine and Physiology in 2013. So with this short introduction, Professor Rothman will today talk on the structural biochemical mechanism of synaptic neurotransmission in the brain, following his lecture on the vesicle transport that he delivered yesterday. Jim, it's a great pleasure to have you here.
After 3 days in a row of hearing you say those nice things about me, I'm beginning to believe you. So, just to remind you, there are a wide variety of vesicle fusion processes that deliver cargoes within the cell for cell growth and division that release mainly protein substances into the endocrine and exocrine circulation to help control metabolism and organ function, and a specialized version of this process called exocytosis that is responsible for the release of neurotransmitters at synapses, whether they involve a neuromuscular junction such as here. This is the end of a nerve cell. These vesicles discovered by Pauli and others contain acetylcholine. And after this nerve fires, it will result in the release of neurotransmitter across the synaptic cleft, in this case to trigger receptors, acetylcholine receptors in the muscle cell membrane to trigger the muscle to contract. And what I'll do today is focus on specifically this aspect of the process of exocytosis or membrane fusion that is closely regulated with the plasma membrane. All of the above processes and many others are mediated by the snare proteins that I talked about last night, and you'll see some examples of that today. But what I'd really like to talk about today are some interesting challenges that we don't really understand at all about specifically synaptic vesicle exocytosis. So they have to do with speed. So a vesicle such as this, a Pauli vesicle, now known to be the COP2 vesicle of Randy Schekman, that buds from the ER, travels over and fuses with the cis Golgi, that whole process might take a minute or so, of which the docking process is probably about half of that. We don't know the exact numbers. But the fusion process itself takes, 10 milliseconds, 20 milliseconds, maybe even sometimes a second. But that's the time constant. It's on the order of, let's say, 1/10 of a second to a second. That's about how fast membrane fusion has to be for this process to succeed in order for cells to grow and maintain their compartments and to release many substances. Now then we have exocrine and endocrine physiology. This happens to be a micrograph of Pauli's of a secretory granule in the exocrine pancreas, storing digestive enzymes. Here it is a moment before you have a meal. Now signals emerge as a result of eating, and this calcium enters the cytoplasm, and it triggers these membranes to fuse. And here, a moment later, might be the same vesicle. It's obviously a different vesicle, now having externalized its content through the process of membrane fusion. Okay? Well, this process also occurs in the same kind of a time range, in the range of 1/10 of a second to a second at the fastest. In the endocrine pancreas, where this vesicle would be instead storing insulin, it's actually a rather faster reaction, but still in the range of tens of milliseconds to maybe 100 milliseconds at the very fastest for the immediately releasable pool of insulin. Now so that is and if you take the snare proteins out of the cell, and as we did yesterday, and put them into lipid bilayers, once they've begun to engage, the fusion process is completed in about the same type of a time range, about 1/10 of a second to a second, in many laboratories under many conditions. So this leaves synaptic transmission in a very unusual position because the vesicles that are closely associated with the membrane, the so-called readily releasable pool, those are the guys intuitively you might imagine fuse first, and then they're replaced by the other ones behind for later rounds of fusion. A vesicle such as this will fuse, as Chuck Stevens discovered in the 1970s, the first one within about 100 microseconds. 100 microseconds. And for synchronous synaptic transmission, which is the type that we rely upon for cognition, that is to say, vesicle fusion that keeps pace and indeed drives momentary neural circuitry, the last vesicle will have fused within 1 millisecond. So the first of these vesicles at 100 or 200 microseconds, the last at a millisecond. This is massively faster. It's thousands of times faster than in any other situation. So it's a very unusual situation from the point of view of molecular mechanism. How can you have a molecular machine, snare pin, that carries out a process on one time scale in most of biology, and in a very important area of biology, neurobiology, carries out essentially the same reaction 1,000 times or more faster. And it's such a simple protein. This is just 4 helices that assemble into a bundle, and it would seem to have only one principle, one molecular mechanism. So how can it operate on such on radically different timescale? So that's the problem that I would like to address today. Specifically, how it is that as calcium ions enter, release occurs in 0.2 to 0.1 millisecond for the readily releasable pool. Now, I want to just remind you of the fact that the presynaptic plasma membrane is also rich with voltage-gated calcium channels. And these calcium channels open up as the electrical impulse, the action potential, reaches the end of the wire. And at the tip of the wire, where the synapse is, so to speak, calcium channels open up and they emit calcium. And you may remember that the basal calcium in the cytoplasm, in most cells, including these cells, is in the low nanomolar range, 10 nanomolar, 20 nanomolar, 50 nanomolar, somewhere in that range. But within a very short time, within a millisecond of the arrival of actually within hundreds of microseconds of the arrival of the action potential, now the calcium rises. Calcium out here is 1 to 2 millimolar. So now, by opening channels, calcium rushes in, and quickly in the region of the vesicle rises to 100, 200, 300 micromolar in concentration. And it is that rise in calcium, as Bernard Katz discovered, that triggers, in one or another way, the release of these vesicles. So what we learned from this is that there must be, and I'll explain to you in a moment what that protein is, a calcium sensor that binds to the calcium. It must somehow, and we know a lot about that calcium sensor through the work especially of Thomas Südhoff and his colleagues. And there must be some mechanism by which that calcium sensor somehow communicates the arrival of calcium to the snare complex, the snare pins that are in that region, to allow the vesicle to fuse, about which we know rather little. So the subject then for today is, number one, how is the information that calcium has entered transmitted in one or another way to the snare complex to enable the vesicle to fuse? And number two, which I will not be able to answer today, but we have some hints, how does this occur in such a rapid time frame as it does? And by the way, if it were any slower, we would be even more stupid as a species than we are. Think about it. How many of you remember what it was like to go to a motion picture? And when they actually had film, okay, right? And then, you know, you would have a bunch of pictures and they would go past the light, right? Now, how fast did they go?
24.
Yeah, it's around 30, something like that. You're not that old, okay, but you remember, okay? He comes from the era when they had slides too, right? Okay, so that's how fast they go. Now, why don't they go faster?
No need.
No need, okay? Because actually the Hollywood moguls wanted to take home their money as profit, okay? So they wanted to have as little film as possible, as little base cost. So you know that they understood psychology, okay? So 30 frames per second is the CPU speed, basically, of our head, okay? Our cognition is operating. We confuse two separate pictures and we fuse them into one. The granularity of our perception of time is basically 30 cycles per second, okay? So they convert that to milliseconds, okay? And that's the frame speed at which our brain is working. Now, you have to go through at least 5 or 10 synaptic cycles to get up to the higher cortices. So when you do the math, if the synapses were slower than a few milliseconds, our CPU speed would be slower. It's kind of interesting to think about it that way, okay? So if synaptic transmission were, for example, 5 times slower than it is, right, then obviously we wouldn't be able to think at the speed that we do think, slow that it may be, okay? Which raises another interesting question, which I might say is a complete digression, but I'll go there anyway, okay, which is, well, if that's so good and we're so smart, why don't we increase our CPU speed 5 or 10-fold? Well, any physiologist here will tell you that the brain already consumes 20 times as much oxygen per gram of tissue and about 20% or something of the blood flow, even though our little noggin is only something like 5 or 10% of our weight. So actually, because every time you work faster, it means you're depolarizing your membranes faster. Most of that energy is used to fuel ATP that fuels the sodium-potassium ATPase. So if we were to, I think actually we're at the fastest CPU speed that, that metabolism actually would allow. So I think that's a fundamental limit. So now we're back to, okay, we now know physiologically what this range of speed has to be. It is in that range of speed. How does it do that? Oh, and there's one other thing that's important in this before I forget. You might actually ask the question, why don't these vesicles automatically fuse with the plasma membrane? I just got done telling you last night that membrane fusion is driven by the assembly of snare proteins, right? And those snare proteins assemble spontaneously, they release a lot of energy, they drive membrane fusion. Well, actually, we have in this synaptic vesicle 70 copies of the V-snare vamp. It's a huge number of copies. And we have in the plasma membrane, the so-called active zone, a nearly pure array of calcium channels and T-snares. So given that, and given that fusion is spontaneous among isolated snares, why don't these vesicles fuse? Well, actually, they do fuse. They fuse very slowly in what's called spontaneous release. And that was the basis for Bernard Katz's discovery of quantal transmission. But the amount of that fusion is really held very, very low. Now, what would happen if in a matter of 10 minutes or a minute or even a second, the time course it takes for insulin to fuse or any other fusion reaction to occur? What would happen if those vesicles fused? Well, that would be it, right? Every thought that you would ever have would be over in that 10 or 100 milliseconds, and there'd be nothing left. Okay? It would be quite an experience, but you would not be able to tell anybody about it. So it's equally as important to prevent the vesicle from fusing as it is to get it to fuse at an unfathomable speed. Okay? That is why it is so challenging and why so many people who are interested in neurotransmission and in biophysics are focused on this problem. Now we have a good, strong hint as to how this works that comes from studies of the state of the snare proteins in the readily releasable pool. And what we know is that they exist in the snare pin state. They're already half zippered. Okay, do you remember yesterday we used the optical tweezer to toggle up and down the snare complex? As far as we can tell from the work of Milton Charlton and his colleagues in Toronto, and also work from a number of other laboratories using other approaches, the snare complex in the readily releasable pool is already half zippered between this being the V snare and this being the T snare, VAMP, Syntax, and SNAP25, between the docked vesicle and the plasma membrane. How do we know that? We know that because when the snare complex is zippered, it is really so tightly wound that proteases can't attack it. It's completely resistant to proteolytic attack. But when it's not yet zippered, it's easily cleaved. It's not even folded, and it's easily cleaved by a protease. So if you could get a protease into the synapse, as Charlton did, and if it happened to be specific for various sequences that are in the end or the C-terminal half, you could then judge whether or not it was this portion, the corresponding portion, was zippered or not. It happens nature has provided us these. Botulinum toxin, for example, tetanus toxin. These toxins which poison synaptic transmission are highly specialized membrane-penetrant toxins that deliver proteases that are absolutely specific for one or another snare complex. That actually is the means by which these pathogens work. They wipe out a snare and they prevent the vesicle from fusing. The readily releasable pool is completely resistant to toxins that address this part of the protein, but completely sensitive to those that address the last part to zipper. So that is the reason that we know that these snare complexes are half zippered. So that helps explain the process, right? Because now it doesn't help us understand why these snare complexes don't complete their zippering. It doesn't help us understand that to fuse spontaneously. But it does help us understand how it can be as fast as it is, at least in part, because they're already half of the assembly of the snare complex. What is in fact the slowest part among isolated snares is already done. It's already complete. So we only have to complete the zippering. Hope that's clear. But this leaves open quite a number of questions. And those are the questions then that I'll focus on with mostly unpublished experiments. How is this half-zippered state, which is intrinsically unstable in the snare pin itself, how is it stabilized in neurons? And the simplest idea would be that there's an additional protein which we've been calling for 2 decades, a clamp, and that this clamp somehow comes in and prevents the further zippering. And the simplest idea would be that that clamp is somehow rapidly removed when the clamp protein binds calcium. I mean, I think you would agree that's the simplest possibility. And then the remaining question, how can the completion of the snare complex's zippering, which normally takes 0.1 to 1 second for a single snare complex, how can this occur 1,000 to 10,000 times faster for the very same species of snares in this context as compared to others? Okay, so those are the questions. Now, there are, of course, other pieces of machinery besides the snare complex that operate in and around the snare complex. There are many, actually, to regulate its assembly and disassembly. The universally conserved component, there is only one, is called an SM protein. It can be called Monk protein or Monk protein. And I won't go into any details about these. They're most thoroughly characterized by Tom Sudhoff and by Richard Scheller. And these proteins act upon the snare complex as privileged chaperones to help the V snare assemble with the T snare in the N-terminal domain, to help accelerate that reaction locally. I won't really have more to say about that, because that happens before the last millisecond. And I'm going to focus now on the last millisecond of leading up to neurotransmitter release. And there are 2 proteins, and as best we can tell, only 2 proteins that come to play. And those proteins are called complexin and synaptotagmin. Both of these proteins were discovered by Thomas Sudhoff. Synaptotagmin, he went on to show, is the calcium sensor of Bernard Katz that actually binds the calciums as they enter and triggers in one or another way the release of the vesicle. Complexin is a simple helical protein that binds to the outside surface of the snare complex. And it has, despite the simplicity of the polypeptide, it has a complex phenotype. But the most interesting, I think, part of the phenotype is what happens when you take this complexin out. So if you remove complexin from the organism, obviously, or maybe not obviously, you don't live, you don't have a brain, but you can get the neurons out in many different organisms and ask what happens. If you take out complexin, the rate of spontaneous release, Katz's minis, goes up over an order of magnitude in most situations. So if you take out complexin, the vesicles that are docked on the plasma membrane, the readily releasable pool, start firing. Okay? In Drosophila, it goes up about 70 times as many vesicles released. Okay? So complexin, in a broad sense, is a clamp that prevents spontaneous release. Now, the other thing that happens is the 0.1 to 1 millisecond synchronous release is now the same amount, the amount of release that would occur is reduced, it's maybe half, the amount of actual net neurotransmission, but it comes out over, guess what, 1/10 of a second to a second. So you lose synchronicity. You release the same number, order of magnitude, same number of vesicles without complexin. They're still calcium dependent, but they're smushed over the better part of a second. So information has been spread out in time. Correlations are lost. In other words, the signal has gone down, the cognitive signal, and the noise level, the spontaneous release, has gone way up. So complexin is controlling signal to noise in an important way. Nonetheless, calcium-dependent release still occurs to a very large degree. If you remove synaptotagmin, there is no neurotransmission, and there is no calcium-dependent release, so synaptotagmin is absolutely required. Synaptotagmin consists of a membrane anchor. It's anchored in the synaptic vesicle, and it has two repeat domains. They're very similar to each other, an A domain and a B domain called C2A and C2B. Each binds calcium ions at aspartic acid residues, and the binding of those ions, especially at the membrane distal B domain, is essential. The A domain can be mutated, and there are effects on synaptic transmission, but none as dramatic as when you mutate out the aspartic acids. And in a classic set of experiments, what Sudhoff did was to mutate these aspartic acid residues and the active site region to increase or decrease the binding constant for calcium, to ratchet it up or down. And what he discovered was the calcium sensitivity of neurotransmitter release was correspondingly ratcheted up and down. And it is because of that experiment that we know that synchronous neurotransmission depends upon calcium binding to these sites. Okay? So that we know. Now, let me also point out again that this protein comes with a synaptic vesicle. There are 15 to 20 copies of synaptotagmin in every synaptic vesicle. The B domain actually docks the vesicle first to the plasma membrane. There's a portion, and I'll say more about this in a moment, called the polybasic region. It's a different site on synaptotagmin B that is not present in A, and it is also separate from the calcium binding site. That positively charged region binds specifically to PIP2, phosphatidylinositol bisphosphate, which, as you may know, is on the inner surface of the plasma membrane and only the plasma membrane. So when the vesicle approaches the plasma membrane, it initially seals the deal by binding to PIP2, and so synaptotagmin, its first role is to act as a tether, essentially to cause adhesion to the membrane. Okay? It appears that that happens even before the snare proteins have begun to engage. Studies from a number of laboratories show that. So now, there's another thing that synaptotagmin does, which is very, very important, which is to say it changes its orientation on the membrane when it binds the calcium. So this shows the B domain only, and this shows the polybasic region of the B domain bound to PIP2 on the plasma membrane. This would, of course, be anchored in a synaptic vesicle. I hope I'm being clear. Shown here are where the calciums will bind. This is a loop that contains the aspartic acids. These are aliphatic residues that flank the loop. What happens is, as discovered by Ed Chapman and colleagues, this molecule rotates 90 degrees when it binds calcium. Calcium binds to these aspartic acids, but also binds mainly to phosphatidylserine negatively charged phospholipids in the plasma membrane. And that actually, when that happens, the synaptotagmin B domain literally reorients 90 degrees. Now, when it does that, it actually inserts these aliphatic residues into the bilayer. We know that that is required for the release of neurotransmitter. Why? Well, if you mutate the calcium binding site, okay, there's no release. If you mutate these residues, there's no release. So it appears that this is the topological switch that somehow allows the vesicle to release. In other words, somehow allows the snare complex to complete. Synaptotagmin is essentially a stupid molecule. It never changes its shape, with or without calcium. It's exactly the same. All it can do is change its positioning. Okay? And in effect, the calcium binding site of synaptotagmin, it's not actually just the protein. It's the protein and membrane phosphatidylserine as a calcium sandwich. Does that make sense? That is the high affinity binding. So this switch is what actually, this little rotation, is apparently what triggers release of neurotransmitter. Now, what I'd like to show you today is our recent experiments that have led us to a completely new and unexpected hypothesis that can explain both how synaptic vesicle fusion is clamped, what is a mechanism that can explain at the same time the inability of these otherwise transiently stable snare pins, what prevents them from zippering to create a readily releasable pool and keep the rate of exocytosis low, spontaneous release, and at the same time can explain how calcium binding by synaptotagmin can trigger the release. And though this mechanism, I'm describing it to you in the context of synaptic transmission, our belief is that exactly the same mechanism underlies most hormone release, most exocrine secretion, release of insulin, and so on. We think it's a broadly conserved mechanism in nature. And the idea couldn't be simpler, okay? Remember what I told you is that everything that comes to membranes has to do, at the end of the day, with a pin, okay, a ball, a hole, and I would just like to add to that a ring, okay, because the idea couldn't be simpler. What if you had a protein that would assemble into a ring to form sort of a spacer or a washer, okay, that would sit in between the vesicle, a little molecular nano ring that would sit between the vesicle and the plasma membrane. And what if the height of that ring was such that it allowed the snare complex to half zipper? And you remember when it completes its zippering, it pulls the membrane closer? But what if it couldn't do that because of this rigid spacer? Well, what would happen would be that the snare complexes would assemble halfway, but they couldn't go any further. You remember this is a binary toggle. I showed you that with the optical tweezers last night. So such a ring would prevent the formation, would prevent the spontaneous release of the neurotransmitter, and it would act as a clamp. Now, what if that ring were composed of the calcium sensor? And what if that ring were to disassemble when it binds calcium? Well, if that happened, then after calcium binds, there's no more ring, there's nothing to prevent the completion of the snare complex, and the vesicle fuses. So that's our hypothesis, that there might be a ring in there. Well, we know what the calcium sensor is, synaptotagmin. We know that synaptotagmin makes its first contact with the plasma membrane by binding to PIP2 using its polybasic region. I've explained that to you. So what happens if we add PIP2, pure lipid, to synaptotagmin cytoplasmic domain AB, pure protein? What happens? Oops. It forms rings. These rings are soluble. The PIP2 that we use in this case is a short chain phospholipid whose critical micelle concentration is above 1 millimolar. We use it at low micromolar concentrations. So these are individual PIP2 molecules. And when a PIP2 molecule binds to a polybasic region, it triggers, apparently, a rigidification of the structure that enables the synaptotagmin to form into a ring. We now know that this requires only the B domain, not the A domain. This is the domain that has the polybasic region, and this is the domain that is the most essential for synaptic transmission. We know that if we mutate the polybasic region, then even if we add PIP2, no polymerization occurs. These rings can be visualized, as you see, on a grid by electron microscopy. They can be isolated as a group, as a class by sedimentation. We can measure the polymerization in a calorimeter and thus get a binding constant and an energy for synaptotagmin B to B. And we can quantify the structure and its shape by fluorescence correlation spectroscopy. And so, this is not 100% yield in solution. It's about a 20 or 25% yield. There's an equilibrium when it's in solution between polymer, closed oligomer, and monomer. What are the size of the rings? They range in size. Here we are between, roughly 30 nanometers plus or minus. From studies that I'll show you in a moment, we can quantify from the diameter. We learn in a moment how they're packed. So we can look at the diameter and say how many synaptotagmins are in there. And the answer is they range from about 15 to 20 synaptotagmins per ring. Okay, so there are 15 to 20 oligomerized in this ring. That's equal to the number of synaptotagmins in a synaptic vesicle. So now what you might be thinking about is that the synaptic vesicle comes up to the plasma membrane. The synaptotagmin binds to PIP2. This then triggers an even more efficient polymerization when the synaptotagmins are localized to each other on or near a membrane surface, and that the ring seals on to the membrane, this is our model, before the snares have even begun to engage. We don't know that for a fact, but it's the simplest view. Okay. Well, when we look, what happens when we add the synaptotagmin AB domain to acidic phospholipid containing monolayers? And I should mention that this is the work of a brilliant postdoctoral fellow, this entire piece of the work, of Jing Wang, who was a postdoctoral fellow in my laboratory until a few months ago, is now working in Basel. Now, when Jing adds the protein to lipid monolayers, you see similar rings and with a similar size. And actually, the two size distributions here refer to soluble rings or the rings that form on monolayers. You see they're very similar in size. But there's a big difference. Do you see how the density, you see how these rings are empty sitting on the grid? You see the same background in the ring or outside the ring. Look at the rings on the monolayer. They exclude stain. They exclude stain because they are actually sucking the monolayer into the ring. You'll see why in a moment. They're actually causing the membrane essentially to pucker up and rise actually above the level of the ring, okay, for reasons that were not clear at first. But clearly there's an interaction with the acidic phospholipids in the membrane. Good. Now, it happens that from time to time, as these rings form, they don't close on themselves. So picture a ring on the surface of the membrane going around and around, sucking up the membrane as it does, because it adheres to the membrane on its inner aspect. Imagine that most of the time it closes off, but sometimes it misses, and it keeps polymerizing. What will it form? It will form a tube, okay? That actually happens. And it especially happens when you use large liposomes and when you add the protein to liposomes. And so what we see are actually rather long tubes, and happily, actually, they can be quite regular. And as a result of being quite regular, they diffract. And as a result of the fact that they diffract, Jing, in collaboration with Charles Sindelar, a professor in the MB&B biophysics department at Yale, have been able to reconstruct from these tubules what the underlying structure of the ring is. So the assumption we're making is that one turn of this helix is essentially the same structure as one ring, okay? And here, we do not have a high resolution structure, but it is high enough resolution for us to orient the B and the A domains within the structure. And what we find is the B domain is up against the membrane. So this is the tubule. This is a cross-section of the tubule. So this would represent one ring. Each of these each of these bumps would represent a B domain. And indeed, we find the B domain up against the membrane. The polybasic region in blue is right up against the membrane. And here's another one and another one, okay? And when we look from the side, it's interesting that the inner aspect of the ring is where the polybasic region is. So that's the region that will bind the acidic phospholipids. And now you can see why the membrane is puckered and sucked up in this fashion. It should be pointed out also that even in his earliest studies of membrane fusion in the late 1950s and 1960s, Pauli pointed out the presence of a consistent pucker on the plasma membrane that could very well be due to this structure. We don't know that yet. Okay, well now having the chance to look at the structure even at a modest resolution, we can see that the calcium binding region is located actually at the interface between two B domains. So that's very interesting because if the site that binds calcium, okay, is located over here at the interface between these B domains, well then obviously when calcium binds, if that site is going to reorient and bind the membrane, then this subunit, the B domain, must rotate into the membrane. It must rotate 90 degrees and quite some distance, okay? So when you think about it, this suggests there's no way for this ring, if the structure is correct, to exist after it binds calcium. It rather suggests that synaptotagmin can toggle between two states. One, in which its calcium binding region is actually formed into the interface between the B subunits to stabilize the ring, and the other where these subunits have rotated and are now inserted into the membrane. So the clear prediction is that this ring should disassemble with calcium. That's exactly what happens. So here we have the number of rings that we see, and here we add calcium, and in the relevant physiological range, 0.1 to 0.5 millimolar, the rings fall apart. If we mutate the calcium binding sites that Tom Südhoff showed were necessary for synaptic transmission, the rings no longer disassemble. In fact, we get more rings. Probably we stabilize more rings because the A domain, which is radially located outside, also is capable of inserting with calcium, and it still has a calcium binding site in this construct, and probably that further anchors the rings and stabilizes them. So to summarize then, the rings form under precisely the conditions that synaptic transmission requiring PIP2, as synaptic transmission does, requiring the acidic region, the polybasic region of the protein as neurotransmission does. And the rings are released or dissociated by calcium, which is, of course, what the proximal signal for triggering release. And the rings are no longer destabilized when the relevant calcium binding site for synaptic transmission is mutated. So the genetics are very strong in and around this ring, though they remain at the moment, up till this point of the talk, the only evidence to connect the ring to synaptic transmission. But we can perhaps do a little bit better because we can look at the interface here and ask what residues are in and around the interface in this suggested structure. And this was done by another postdoctoral fellow, Oscar Bello, in collaboration with Jing-Wang. And what Oscar did was systematically change each and every one of the residues that was anywhere in the vicinity of what was proposed to be the contact region between the two B domains, on the one side of the B and on the other side of the B. And we thought if our hypothesis was correct, if our structure was right, sooner or later we should hit some residues that are required for that protein-protein contact. And indeed, some of the residues on each of the surfaces of the B domain that were mutated had an effect. And they are shown here. For example, leucine 307 and proline 310 on one face of the protein, and on the complementary face, residue 349 phenylalanine to alanine. When these are mutated, rings no longer form, but synaptotagmin retains each of its additional functions. It still binds to PIP2, okay? It still inserts its flanking hydrophobic residues with calcium into a bilayer. So it still undergoes that toggle to release neurotransmitter. The only thing it doesn't do, as far as we can tell, is form rings. Now, this set us on a path then to making mutations and putting in the synaptotagmin that would destabilize the ring, and to try to put them back into neurons and see what the effect might be. Is there an effect of forming this ring in neurons? And as we were starting this, we actually, as I said, smart is good, but lucky is better. A syndrome in humans was described in which the protein, the very protein we are studying, synaptotagmin, is mutated in these families. And the families have an interesting neurological syndrome that broadly falls into the category of Lambert-Eaton myasthenic syndrome, in which there is chronic and often, but sometimes intermittent weakness in muscle. And when biopsy is done and when they're studied electrophysiologically, they have a poopy neuromuscular junction is really what it boils down to, okay? And it tires easily. And these patients are mutated in synaptotagmin 2. Now, I have been telling you all about synaptotagmin 1, and I've been sparing you of the fact that there are 17 or more synaptotagmins, okay? Synaptotagmin 1 and 2 are the two principal neurotransmitters, as I understand it, for synchronous neurotransmitter release in the brain. And in particular, synaptotagmin 2 is important for the neuromuscular junction. So you would think that that this might be, in general, if you have a family with this sort of a syndrome, it's obvious that this allele that they carry is not going to be all or none. It's going to be a graded loss of function. Otherwise, they would be dead. But it's also interesting, and I point out that it's autosomal dominant. Okay? I'll tell you why that's interesting in a minute. So Oscar decided to test this protein, P310N. Now, actually, for those of you with photographic memory, which does not include me, this happens to be a residue that he had located independently at the interface. And when that's changed to leucine, there is no ring. When nature, in this unfortunate family, changed it to an asparagine, there are less rings. And the rings that we have are actually less complete, typically. So this explains to a large degree the disease and gives us confidence that the formation of this ring is likely to be an important physiologically in regulating neurotransmitter release. Now, you remember that it's an autosomal dominant disease. What would happen if you put a bad version of synaptotagmin into this ring? Let's imagine that we have a mutant where we take one of the, the B domain has two faces that interact with each other. If I mutate one side and I add it into a bunch of synaptotagmins, the synaptotagmin chain will grow, but it will terminate when this bad subunit adds. So it'll be like a chain terminating mutation. And in fact, what Oscar has found is if you take mixtures of his mutant synaptotagmins that don't form rings, you add them with the normal ones, and now you ask what happens, you get truncated rings that we can see in the electron microscope. Sort of like C-shaped structures that don't go all the way around. That explains, of course, why this is a dominant disease. If you cripple the ring by putting in one or more bad subunits, it will lose its integrity and will likely be less of a barrier to the assembly of these snare complexes. Now, what would you predict would happen then if you had no rings or if you had unstable rings? you would expect that the rate of spontaneous release of neurotransmitters should go up, right? Because we're fundamentally proposing that the ring acts as a clamp, okay? Is that really true? So we've begun to test this in collaboration with colleagues at UCL in neurons, but we don't have our first results yet. But we do have a poor man's version of the neuron called the PC12 cell that we're capable of working on at Yale. And this is the work of Warden G1, another postdoctoral fellow. And these cells are deleted for synaptotagmin 1. They, however, have other synaptotagmins in them that are present. When he what he's measuring here are several things. He's measuring the number of vesicles containing the number of vesicle fusion events, let's just start with this, that occur spontaneously. That means before depolarizing the cells. And he's measuring the number of vesicles that are docked to the plasma membrane. And this has been done, the number of docked vesicles has been measured both by turf optical microscopy and also by electron microscope sectioning. So we have very similar numbers. These happen to be the turf numbers. So whether you have no plasmid or you put in the normal synaptotagmin and it's overexpressed, or you put in this mutant that doesn't form rings, any one of them, you will get about the same number of docked vesicles. But the rate of spontaneous release is drastically increased when you have the synaptotagmin mutant. So that suggests that, the ring incompetent protein is acting to prevent an endogenous clamp. Now this is a somewhat imperfect experiment, but it's not impossible to interpret. These cells have a low spontaneous release, we believe, because they have synaptotagmins 7 and 9 expressed in abundance. And that clamps them down. But when we put in the mutant that dominantly interferes, remember autosomal dominant, that interferes with all the synaptotagmin rings that are present and allows release to occur, spontaneous release. We're now in the process of trying to clean the experiment up by using siRNA to take out the other 2 remaining synaptotagmins. But I believe it's probably a reasonable interpretation. Now, how general are synaptotagmin ring oligomers? Well, as I've mentioned, there are a large number of synaptotagmins. We haven't tested all of these, but we've tested some of them. And here is synaptotagmin 1. Synaptotagmin 2, of course, forms rings. Synaptotagmin 7 forms rings. Another soluble protein that is a regulator of exocytosis, DOCK2B, forms rings. So it seems to be a very general property of synaptotagmins. We thought we would try another very interesting synaptotagmin called the extended synaptotagmins. Extended synaptotagmins are not involved in vesicle fusion, rather the opposite. They are the key ingredient in forming cortical endoplasmic reticulum. This is endoplasmic reticulum that in a regulated fashion adheres to the plasma membrane to allow intimate exchange of materials such as phosphoinositides between the 2 membranes. And that synaptotagmin is needed is rooted in the endoplasmic reticulum. It binds PIP2, typically, in the plasma membrane. And there are several species of extended synaptotagmin, but in one or another way, it's not known exactly how, causes the adhesion to the plasma membrane. Remarkably enough, extended synaptotagmin also forms rings, and these rings tend, unlike the others, to form networks, which suggests a mechanism for how they might adhere. So it's a very general property of synaptotagmins to form rings. And this, in turn, suggests a possible evolutionary origin of exocytosis that I'd like you to consider. Yeast have membrane fusion, of course. They have snare proteins galore and intracellular protein transport. They do not, however, have any regulated exocytosis. They do not have calcium-triggered membrane fusion. They do not have secretory, though they secrete, they do not, the vesicles constitutively fuse. They do not have stored vesicles that fuse upon command. They do not have actually synaptotagmin. They have snares, and they do not have synaptotagmin as it is defined as a vesicle protein. But they do have extended synaptotagmins, and they do have cortical ER. So you have in yeast both of the working parts of what we would argue is the fundamental apparatus of regulated exocytosis, including at the synapse. They have snare pins, and the snare pin's job is to irreversibly adhere membranes, causing fusion, inexorably causing fusion once they have begun to engage. This is the fusion engine. Then they also have synaptotagmin rings. Their synaptotagmin rings are not even located in vesicles. They're located in the endoplasmic reticulum to form spacers, close apposition with the plasma membrane. The job of those synaptotagmin rings is the exact opposite of membrane fusion. It is to allow two membranes to come within molecular contact distance, but never to fuse, so they can be later taken apart when the ring disassembles. So the goal of synaptotagmin rings, I would argue fundamentally, is reversible adhesion preventing fusion. When these two principles are combined in evolution, then you automatically generate calcium-regulated exocytosis. And it is easy to imagine how that could happen. All you need to do is relocate an extended synaptotagmin to a transport vesicle, and according to the simple mechanism that we suggest, it will automatically happen. It will not be optimized, but it will automatically happen. So what are the outstanding questions then that we can return to, and what are some of the answers that we've achieved and what remains? Okay, how is the half-zippered state stabilized at the neuromuscular junction, at synapses generally, and perhaps for exocytosis universally? It is through rings of synaptotagmin in all likelihood that form at the interface. How is it in the special case of synaptic transmission that these snare proteins can complete their fusion within a millisecond, 100 microseconds to a millisecond, whereas they're thousands of times slower in every other case? Well, I mentioned complexin, and I won't say anything further about complexin, except that complexin is obviously key to further energizing the synaptotagmin ring structure in some way or other that allows fusion to be completed very rapidly. In the absence of complexin, synaptotagmin alone, I believe, can explain clamping and release on the timescale of exocytosis generally, but not on the timescale of synaptic transmission. So complexin now needs to enter the picture in a forceful way. But I would also suggest that part of the answer may come from the fact that a ring is likely to engage and recruit as many as 15 to 20 snare pins. Now, when I suggested this very simple evolutionary model, that assumes that there's no adhesion between the snares and the synaptotagmins, right? I mean, in the simplest world, the ring and the snares completely ignore each other, and that might be enough to prevent these guys from powering through and fusing. But in evolution, of course, it would be much cleverer for the snare complex to bind the synaptotagmin protomer and therefore perhaps be held in a configuration even less prone to fuse, and to ensure that the snare complex is assembled and recruited. And possibly, since there are 15 to 20 synaptotagmins in the ring, to recruit as many as that number of snare pins, to allow many snare pins to cooperate. And perhaps it's that cooperativity of a large number of snare pins that is important for making fusion that much faster. So it's hard to test that experimentally at this stage. So we engaged in a simulation. It's what's called a coarse-grain, quite realistic simulation that will be published in the PNAS in collaboration with Benjamin O'Shaughnessy and his colleagues at Columbia. And what this simulation assumes is we start with N snare pins, half assembled. We form them at the in the computer at the interface between a realistic size vesicle and a realistic size membrane, and we allow them to be placed randomly okay, consistent with that geometry. We take into account the known energies and kinetics of zip ring. We take into account the electrostatics, the entropic factors, and many other factors. So it's a pretty realistic simulation. And we let them go. And we ask, over time, how do they arrange themselves? Guess what? They arrange themselves automatically into a ring because they start jostling each other entropically and arrange themselves into a dynamic but well-spaced ring. That ring creates pressure at the contact point, and that eventually transits the activation energy for membrane fusion. We can, in the simulation, start the clock and measure how long does it take, how long do you have to wait before the first snare complex zippers out of that large number as a function of how many we started with. And there's actually an exponential relationship of waiting time. And on the basis of this factor alone, starting with the idea that a single snare pin fuses, takes 1 second to complete, okay? Actually, the experimental data is variable between labs, somewhere between 50 milliseconds and 500, so we're actually being very conservative here. It would take in the range of 5 to 10 milliseconds for 10 snare pins for the first of 10 to complete, and progressively less time for even more. We didn't run the simulations for more. So the long and the short of this is, merely having dozens of snare pins in the vicinity and getting rid of their impediment to zip ring might be sufficient to go a long way to allowing the rapidity of fusion that's occurred. If one of them is too slow, if you have n of them, they'll be exponentially faster. That's what comes out of this. But I personally don't believe that it's enough because complexin is vitally important. So then, what are the updated outstanding questions that we can go forward with? Most importantly, do synaptotagmin rings in fact form at docked synaptic vesicles in vivo? I actually haven't shown you a shred of evidence that these rings actually exist in a cell. Never mind a neuron. We have a growing mountain of genetic evidence on all sides of that, but still no direct evidence. And we're working very hard to use pushing super resolution microscopy to the absolute limit to see if we can visualize 30 nanometer rings. I believe that we will because we've succeeded just in the last several weeks in visualizing a donut shape with a DNA ring, or DNA origami ring of 30 nanometers. So if they're there, we will see them in the fullness of time. How do half-zippered snare pins interact with the synaptotagmin rings? Well, as I've told you, it's unlikely they ignore each other. And finally, how does complexin synergize to trigger rapid release? A critical advance was made in the last months by Axel Brunger at Stanford, who solved the first single crystal structure of synaptotagmin bound to the snare complex. It's been known for years that a single molecule of synaptotagmin, the A-B domain, will bind to the snare complex. And it's been known for several years that it is the B domain specifically that engages in this contact, the domain that's most important, and that it binds to 2 portions of the SNAP25 helical face of the snare complex. That's also been known. In the crystal structure, among other contacts, there is this contact between the B domain and SNAP25, predicted to be the strongest contact. And indeed, mutating residues in this region partially compromises synaptic transmission kinetics. So this is likely to be a relevant contact. There are probably other contacts, frankly, that we've seen on membrane-bound complexes by 2D crystallography. We don't think this is anywhere near the whole story. We think it's more likely there's choreography in which a number of contacts are used successively. But there's no doubt that synaptotagmin binds to the snare complex in a definite way, and that's what I wanted to convey to you. And we can be fanciful, and we can take our rings, looking from the top, and we can model in the coordinates, just simply drop in the snare complex based on Axel Brunger's structure. Maybe more revealingly, we can look at it from the side. And it's easy to begin to see how a ring could restrain the snare complex, maybe in a more particular way, from completing its zippery, maybe by holding on to it. We, of course, don't know that this model is correct. I can almost guarantee you, actually, it's incorrect. Because I have a pretty long track record of proposing models, every one of which turns out to be incorrect at a certain level of detail. But the other thing I can almost assure you is that something as beautiful as a ring structure like this is likely to be correct in its generality. And so I think over in the fullness of time, in the work from many laboratories, hopefully including ours, we will come to a detailed structural understanding of how rings of synaptotagmin, potentially rings of other regulatory proteins interacting with those rings, and the snare proteins and others, how they conspire to create a highly energized state from which what can only be described as explosive release occurs. And my personal belief is that when you have a release process involving membranes that can occur in 100 microseconds, it's certainly too fast for enzymology. It's too fast, actually, for protein assemblies to form. It's not too fast for example, a synaptotagmin to rotate on the membrane. It is too fast for the synaptotagmin ring to dissociate and diffuse away. I believe that we are dealing probably in a range of physics, which is more like an explosion, in which there is a highly energized substrate that is released, and in an explosive way, the membranes are merged. That's my personal belief. We'll see how things develop over time. So there's a lot more to understand. I'm fortunate to have a number of very capable colleagues. Sham Krishnakumar, who is a senior research scientist who has been working with me for several years and who with me is leading this project. Jing Wang, who discovered the rings and has now gone on to her just rewards. Oscar Bello, still a postdoctoral fellow. Feng Li, who's done important work on the rings and the biochemical studies involved in studying them as soluble entities. Alexander Rebane, who with Young Li Zhang has done the optical tweezer studies. Maria Zanetti, another postdoctoral fellow who characterized the various other synaptotagmins that form rings. Sarah and Jeff are contributing technologically to many aspects of the project. Warden, who has done the work on the physiology of the ring that suggested that the ring actually regulates exocytosis functionally. And various collaborators, I've mentioned Ben O'Shaughnessy already, Young Lee, faculty member who does the optical tweezer work, Chuck Sindelar, with whom all of our current and past EM crystallography and EM analysis is being done. Frederic Poncet, with whom I've had a long-term collaboration in Paris. He's a professor of physics at École Normale Supérieure. And we collaborate extensively on the biophysics of snare assembly. And David Baddeley, an assistant professor at Yale, whom we are hunting down the rings with using super resolution microscopy. So thank you all for your attention, many of you over 3 days.
Thank you, Jim, for another exciting lecture. There is a possibility to ask questions. I'll bring the mic over.
Thank you. Is it possible that the rings might also enrich the lipids underneath to change, or so that, because besides having more hairpins to apply more force, could you possibly have a membrane structure which is weaker and doesn't require as much force to cause the fusion?
That's perfectly possible. It may even be likely. PIP2 exists in the plasma membrane in nanodomains. The exact size is maybe 50 to 100 nanometers. I may not be recalling exactly. The T-snare subunit syntaxin, as Reinhard Jahn found, its juxtamembrane region, which has lots of positive charges, actually produces PIP2 clusters, and therefore it co-clusters with PIP2. So you have actually clusters, nanoclusters of syntaxins and therefore T-snares, which also Reinhard and his colleagues have demonstrated even in vivo. So underneath the synaptic vesicle lies a nanopatch larger than the vesicle itself, but certainly of the same order of size, in which the local concentration of PIP2 has variably been estimated to be between 10 mole percent and nearly 100 mole percent on the cytoplasmic surface. We don't know the exact number. The T-snares are highly clustered in that region. So the physics of that region is likely to be very different. And it's likely to be more prone to curvature for the very reason that there are repulsive negative charges. It's to this very region that synaptotagmin is likely to target and around which this ring is likely to form. So there's a lot that remains to be learned, but there are some hints of levels of sophistication and complexity. This is going to be one of the most finely tuned machines in nature. There is no machine that is more fundamental to animal existence than this. And there is no machine within an animal that needs to be more tightly wound, more tightly regulated, and more energized than this machine. So it's going to be a very, very complex machine. But I believe at the end of the day, it will have a simple symmetrical structure, because there must be an enormous amount of force. Every snare complex is generating tens of piconewtons of force, times 15 to 20, okay? Add in the binding constants for complexin, et cetera, synaptotagmin. The amount of pressure that is being placed at the contact point. You know, I don't want to be like Donald Trump and say it's huge, okay? But it's huge, okay? It's 100 times the atmosphere or more. It's huge, okay? And so why doesn't this thing fall apart? Its forces must be internally balanced as in a beautifully architected cathedral, so that it only takes the slightest introduction of asymmetry to release sufficient force. And I believe that's the secret to explosive release. If that's correct, there will be a beautifully symmetrical structure, even at the highest level of force.
There were a couple of more questions. First here and then Nick.
Last night you briefly touched upon your early work where you showed the NEM allows the vesicles to dock, but it prevents fusion. And transplanting that into your present-day work where you show that these rings are dissociated by calcium, have you added NEM to see what happens to these ring structures, whether they dissolve or they sensitive?
NEM does, to my knowledge, NEM does nothing to that ring. And the NSF comes into play after the fusion reaction to recycle the snares.
We have one question here.
How and at which stage is the neurotransmitter actually accumulated, concentrated inside the vesicle?
So that's a good question. My own work doesn't concern that, but it's quite well understood. As you might imagine, there's a transporter that is within the wall of the synaptic vesicle. And depending on the type of nerve, if it's one that secretes acetylcholine, it'll be taking up acetylcholine. If it secretes glutamate or GABA or any other neurotransmitter, there'll be a dedicated transporter. The transporter is a proton antiporter, so a proton inside the synaptic vesicle will exit one proton or so for every, I think it's one, but in any case, a stoichiometric relation for every co-transported neurotransmitter. And so, and in turn, the inside of the synaptic vesicle is maintained at an acidic pH. Typically, it's in the pH range of 5 as compared to the 7 for the cytoplasm. And there is a vacuolar ATPase that sits on the outer surface of the synaptic vesicle that hydrolyzes ATP in the cytoplasm and pumps the proton in that energizes through its efflux the countertransport a la Peter Mitchell of the neurotransmitter. So that's how that works.
Jim, maybe one more question. So maybe I missed it at the beginning of your talk, but this incredibly high speed of fusion that takes place at synaptic junctions doesn't seem to agree with otherwise slow CPU of the brain. So at which point physiologically is it critical to have that high speed microseconds?
Well, no. The first one comes out in 100 or 200 microseconds. The last one comes out in a millisecond or so, maybe 2 milliseconds. And again, I'm not an expert on this, but the time it takes for synaptic transmission overall is typically several milliseconds, maybe 5 milliseconds at the most. Of that, you already use up roughly a millisecond for the channel to open and for the calcium to diffuse, and something in that range. I'm sure I'm off a little bit, but it's something in that range. Then the next millisecond, let's say, is for the vesicle to open the hole. Then it takes roughly a millisecond for the neurotransmitter to drain out and diffuse across the cleft. Then it takes another, whatever, millisecond for it to bind the receptor and the receptor to open a channel, and then the ions need to come in there. So when you add up all the little bits and pieces, if any one of them was several times slower, the overall speed would be compromised, and most importantly, the synchrony. Because, and this is just my, I'm not a neuroscientist really, but it's just my, my amateurish way of thinking about it, is that, for example, when you asked me that question, I noticed that your voice came in at the same time as I saw your lips moving. Okay, it would be kind of a bad thing if they weren't
Hopefully.
synced. Okay, like in a bad spaghetti western with Clint Eastwood from the 1960s. So, why does that happen? It happens because the information comes in through separate routes, but it's being passed in synchrony to the higher associative cortices. Okay, so even a slight spread in that will cause a lack of clarity. Okay, such as we may experience at the banquet tonight.
Correct.
Okay.
Any last-minute questions?
Good. Thank you.
If not, let's thank Jim Rothman once again.